Hello everyone! In this presentation I will talk about the role of genetics to deliver food security. First, let's define food security. According to FAO, food security is achieved when everyone has access to sufficient, safe and nutritious food to lead a healthy life. However, the future reliability of current food system is severely compromised by two factors. First, climate change, which with extreme weather events set to become more severe and more frequent, it has been identified as the primary determinant of food insecurity. Secondly, the steadily increase in world population, forecast to reach 9 billion of inhabitants by 2050, is expected to exert further pressure on stable resources and overall availability of food. In this scenario, how can we cope with oncoming challenges posed by global ecological issues? Since Neolithic, humans have been reacting to natural diversity by selective crossing of species carrying desired traits into the process of domestication. With domestication of plants, humans began to manipulate the food to satisfy their needs. In this way, we have generated seedless bananas, larger maize grains, long carrots, tastier melon, and so on. With scientific progress, we passed from empirical selection of food to the use of the chemical components, radiation, until to manipulate precisely the DNA, simple base plotting means already used in nature, biological processes such as cloning, as well as bacteria and viruses. We have created species capable to produce own pesticides, enriched in vitamin supplement and disease resistant, to preserve food and feed the population. We can assert that the precision and the reliability of this process is due to our knowledge on the mechanism of the DNA transmission. Briefly, the DNA is a molecule holding all instructions which every organism needs to survive and reproduce. Such source of information is highly organized in fragments called genes. In a cell, all genes are highly compacted in a structure named chromosomes. At chromosome level, genes are naturally reshuffled and transferred through a process called meiosis. In detail, meiosis is a specialized cell division which aims to produce fertile gametes, sperm and ovum in human, pollen and ovule in plants, by reducing the chromosome set by half. During meiosis, maternal-parental chromosomes align and genes between chromosomes are exchanged, creating new combination of genes similarly to a car shuffle before starting another round. In genetic terms, such recombination passes to another round of cell division and gives rise to variation in the offspring, otherwise we would all be clones of ourselves. However, in cross species such as wheat, such process has suffered the effect of domestication. Wheat is an essential component of global food security system, providing nearly one-fifth of the total calories of world's population. Common wheat arose approximately 10,000 years ago from two hybridization events. The first brought together the wild goat grass Triticum urartu and the species related to Aegilospeltoides to form pasta wheat, which underwent a second hybridization event in which it was combined with another wild goat grass, Aegilosptauski, to produce bread wheat. As a result, we have originated a wheat carrying more than one set of the chromosomes, one from the parent and another one derived from the wild progenitors, a condition called polyploidy. Related chromosomes may potentially be engaged during the meiotic recombination in addition to parental chromosomes. If parental relatives' chromosomes fail to recognize each other, the genetic exchange cannot take place and we observe cells having unbalanced chromosome number, unable to produce fertile offspring. Such situation results in huge yield losses for breeder companies. In addition, recombination events occur at low frequency and are distal along the chromosome. Therefore, the majority of genes of interest rarely recombine and are inaccessible to breeders. Thus, investigating the mechanism involved in wheat to control the correct chromosome pairing during meiosis is my project aim. My approach is to cross wheat treated with AMS, ethyl methanosulfonate, a chemical mutagen which induces a mutation in a gene of interest, in my case in a meiotic gene, screen and analyze mutant plants from a fluorescent microscopy. How do chromosomes behave if we reduce the amount of a meiotic gene by using a chemical mutation? If successful, is it possible to use these mutant plants to introduce traits of agronomical interest from a relative species into the wheat? Answering this question will be my goal. To conclude, these studies, besides enriching the knowledge of this process, may enable breeders to enhance the genetic variability by begetting novel varieties which selectively adapt to the oncoming environmental changes and ultimately equip us with a strategy to achieve food security. Thanks to all, and if you are still curious, take a look to the web link of my research project network.